Welcome everyone, and welcome to this video dedicated to the infrared spectroscopy of proteins. The aim of this video is to give you an overview of the use of infrared spectroscopy to study proteins, both structural studies and also functional studies. And we will also be looking at the use of the different amide bonds in the infrared spectra to perform those studies. The video is based on a couple of resources, mostly Barth's and Kong and Yu's reviews. I would suggest that all the resources are available from the Blackboard for the module. I would suggest that you start from Kong and Yu's resource. It's the most readable, it is shorter and most readable of both reviews. And then you move on to Barth's review. They're both from 2007. And additionally, I have obtained some small sections from this thesis by Lee in 2016. And I will highlight throughout the presentation where those bits are. So, what can we do with infrared spectroscopy of proteins? Well, infrared spectroscopy allows us to study a protein's structure, and we can also study the reactivity. We can look at how proteins fold, unfold, and misfold. And we can also use infrared spectroscopy to study larger biological systems, for example, bacterial strains, or even to differentiate and classify microorganisms. So it's quite a really useful technique, and it has many advantages. It has a large application range. You can look at small peptide fragments or small proteins to very large ones, very large proteins part of membranes. It has a high time resolution, so you can study reactions that happen very fast, in very short periods of time. It has typically a short measuring time, which is good to perform the studies quickly. And you also, in generally, need a very small amount of sample, 10 to 100 micrograms. Also, the cost of an infrared spectrometer is much lower than other equipment typically used to study proteins, for example, an NMR. You cannot deduce the structure of a protein exclusively from the infrared spectrum. There are too many bands. However, what you can really observe is changes in the structure. For example, because of the protonation of side change, you can follow phosphorylations, you can follow enzyme activity, because you can detect the changes in the protein's structure before and after the reaction takes place, or protonation, for example. And I will be looking later in the video at exchange, hydrogen deuterium exchanges, for example, as a way of observing, of studying a protein's reactivity. You can obtain information about the redox state of the protein because you are altering the electronic distribution of the protein, and then that alters its vibrational spectrum, which is what the infrared spectrometer detects. You can look at bond parameters, the strength, length, the distortion, angles, the conformation of those bonds. Hydrogen bonding. Typically, hydrogen bonds will lower the frequency of the stretching vibrations and will increase the frequency of bending vibrations. So you can look at hydrogen bonding in molecules, in proteins, by infrared spectroscopy. And you can also look at the electric field and how it changes, for example, during a reaction, if you're studying enzyme activity. Looking at the width of the bands in an infrared spectrum of a protein, you can get information about the protein's conformers. The position of a band is typically slightly different when, for each conformer, because the structure, the way that protein is distributed spatially changes, and that changes the vibrations of the bonds, and that means bands become broader. So flexible structures, which with many conformers, will tend to give broader bands, and more rigid structures, more narrow, more sharp bands. So the bandwidth, and there are ways to calculate that mathematically, is a measure of the conformational freedom of your protein. And you can observe that when performing a reaction, when a molecule binds to the proteins, typically that will restrict the freedom of the conformational freedom of the protein, and you will be able to observe that by infrared spectroscopy. One thing you can look at is the absorption of the amino acid side chains. So we are not looking at the backbone, we're looking at the side chains. And this is a table from Barth's review, in which it gives you an example of the different functional groups in amino acid side chains, and typically where they appear, and some comments about how they appear. So as you can see, it's a way of following what happens to the amino acid side chains of your protein. The most interesting part of the infrared spectrum of a protein is the bands related to the amide vibrations. And you have quite a few amide modes of different usefulness. We will be focusing mostly exclusively in amide 1 and amide 2, but there are quite a few of them. The first ones here in this slide are amide A and B. And those are related to the NH, to the amino group in the proteins, and it is quite insensitive to the polypeptide backbone. It depends on the strength of the hydrogen's bond form, so it can be useful to determine that strength of hydrogen bonds. The most important component is the amide A band, and then it has a second component which is much weaker, which is called amide B mode. Another absorption important in proteins is the amide 1 mode, and this is by far, with amide 2 that I will discuss later, the most useful of all the amide bonds, so everything of what you can observe in an infrared spectrum of a protein. It is mainly due to the carbonyl stretching vibrations, with some minor contributions from other groups, and it can be really useful to determine the structure of the protein, and especially useful to determine the secondary structure of the protein, and it's widely used in secondary structure analysis. It's also because although it is mainly due to the carbonyl stretching vibration, it has some contribution of the NH in plane band. It is sensitive to the deuteration of the nitrogens of the amino groups of the backbone. So it can also be using in the deuteration in high studies, and I have a few examples later on in the video. It is very insensitive to the nature of the side chains, so it is useful specifically to see the structure, the backbone of the protein, and especially the secondary structure. It has a small problem, which is that it appears roughly at the same wave number as the OH bending vibration from water molecules. So typically, or the sample is prepared in larger concentration, so the amide 1 band is very obvious with respect to the OH bending vibration from the water present in the sample, or the samples are prepared in deuterated water that has a bending vibration of the oxygen deuterium bond in another area, so there is no overlap with the amide 1 band. A large splitting of the amide 1 band is typical of beta sheet structures. So it suddenly splits in two, and there's a main band in the 1630 inverse centimeters, and a side band, a smaller one, at around 1690 inverse centimeters. And for beta sheets, the hydrogen bonding will affect the position of this main band, and less the side one. The hydrogen bonding can also be observed in alpha helices, and when you have solvated helices, that when you have helices that are forming hydrogen bonds with the solvent present, then that will lower the wave number to which those bands appear. So here, for example, this is a table again from Barth 2007, and it gives you an idea of where, depending on the structure of the secondary structure of the protein, roughly where you know, this is bands in water, and it would be bands in deuterated, if you do the experiment in deuterated water. So typically where they appear, and roughly the range that they move. And as you can see, it is different if it's an alpha helix or a beta sheets, or if it returns, or if it's a section, you are looking at a section of the protein which is disordered, and you can follow that using infrared spectroscopy. Amide 2 is also quite useful. The amide 2 bond appears around 1550 inverse centimeters, and it's a combination of a couple of vibrations, the NH in plane bend and the carbon nitrogen stretching vibrations, with some smaller contributions of other bonds. It is hardly affected by side chain vibrations, so again it gives you a good idea of the backbone of your protein. And it's slightly more difficult to interpret. It has more contributions. It can be useful to look at when performing deuterating studies on deuteration of the amino groups. Because it changes the position of the band quite dramatically, and it is not observed, or is barely observed, if you record the Raman spectrum of your protein rather than the infrared spectrum. The amide 3 is less useful, although it can be used. There are some studies in that it is used in combination with amide 1 and 2 modes in secondary structure prediction, and there are quite a few contributions of the various bonds, and it's also influenced by the structure of the side chain, which means that it is more difficult to interpret. It gives you less specific information on the backbone, structural backbone of the protein. There are other amide bonds of less usefulness, 4 to 7, depend on various bending modes, and even one around 200 inverse centimeters that is looking at the torsion of the protein skeleton. But in general, amide 1 and 2 are by far the most useful ones. And all the examples I'm going to give you on how to use infrared spectroscopy to study protein form and function are based on those two modes. So what can you do with infrared spectroscopy to study proteins? Well, it does have a high time resolution, less than 1 microsecond, so it can be used to study reactions that happen very fast. It can, again, as I mentioned before, from small soluble proteins or peptide fragments to large membrane proteins. It's so sensitive that it can detect changes in the environment in a specific atom. So it is really quite useful to measure activity, for example, enzyme activity measurements, or to pinpoint how the structure of the molecule, of sections of the molecule down to a single atom, influence or how they change with when a reaction is performed. It can also be useful to follow water, the interaction of water with proteins, and the hydration of the protein structure. If you are interested in reading this, there is section 2.4.2 of Lee's thesis that covers this into more detail. It does have its difficulties, and one is that the bands are broad, and as such the infrared spectra, they are not easy to interpret, or it is difficult to extract information from them, because look at, for example, this spectrum of a CM receptor protein. And look at the amide 1 band that appears roughly between 1700 and 1600. So it is a band. It's difficult to observe if there's happening there, if anything is happening there. It's or how that band would change after a reaction. However, there are mathematical analysis methods that you can use to enhance that, computational methods to enhance that band. And if you look at the image below, it's the same. Can you see between 1700 and 1600 is the same range? So that's the amide one, but instead of being the Fourier transform infrared spectrum, it is the second derivative spectrum. And as you can see, what in the original spectrum is just a band, it actually appears as several bands and shoulders, and those are easier to, well, they're easier to see. And then when a reaction takes place, hydration, deuteration, and enzyme activity, it's easier to see how those peaks change and move according to the reaction. So it's not so much the spectrum, the raw spectrum, the original spectrum, but the treatment of that spectrum that will give you the information. This is, for example, how you can use amide bands to determine not only the nature of the secondary structure of the protein, but also the proportion. The area under each peak corresponds to the proportion, the percentage of, you know, beta turns and alpha helix and beta sheets in that particular protein. So you can use and this again is the second derivative. It's not the spectrum. It's not the original spectrum, it's the second derivative. And all these bands correspond to amide 1 bands. They are all between, you know, the 1700 to 1600 range. So you can use amide 1 band very effectively, and also amide 2, to work out the proportion of the different types of secondary structure that you have in your protein, because the area under the peaks will reflect, the peaks themselves will reflect what structures you've got, and the area under the peaks, the percentage of your protein that has that particular structure. Another useful thing that you can do with infrared spectroscopy is to perform hydrogen deuterium exchanges to see how the amide proton, the formation of hydrogen bonds, the changes of the hydrogen bonds to experimental conditions, or to the presence, for example, it's an enzyme, to the presence of the substrate, because it will reflect the dynamic of the proteins, how it is moving. It is sensitive to the secondary structure, so if there are changes in that secondary structure, they will be reflected by doing a hydrogen deuterium exchange. And also things like pH conditions, temperature and pressure, but specifically pH has been very useful, will reflect on those bands. So for example, here you've got in the top the original spectrum and in the bottom the second derivative. In the study of the amide band, look, the range is between 1600 and 1700. So how amide 1 band in pyruvate kinase reacts in a hydrogen deuterium exchange. And as you can see, the different colors reflect different times during the reaction, from 1 minute to 60 minutes, and the black line reflects performing the reaction in deuterated solution, and the arrows show how the bands increase or decrease as the time passes. So again, you can study both experimental conditions and the behavior of that amide proton, and the structural behavior of the protein in the presence of water, solvents, pH changes, or other chemicals. For example, a substrate, if you are looking at an enzyme behavioral activity study. Another useful thing you can do with amide bands, amide modes, and infrared spectroscopy is something called reaction-induced infrared difference spectroscopy. And it is used to investigate molecular mechanisms of protein reactions. So the relationship between the reaction taking place, again think of enzyme activity, and the structural changes that take place once the reaction happens. So in this spectrum here on the top, you've got imagine a protein in a particular condition, state 1, before the reaction takes place, and a spectrum is recorded. And then the reaction happens. It could be a proton exchange, it could be, if it's an enzyme, the presence of a substrate. And the reaction takes place, and then you take another infrared spectrum in the second state after the reaction has taken place. And if you plot both spectrum, well, you might be able to observe that yes, the two lines don't quite overlap. There might be some changes, but it's not something that you can see straight away in the original spectrum. This would be an absorbance spectrum, not a transmittance spectrum. However, if you do the difference spectrum, if you subtract spectrum 2 final protein from spectrum 1, the initial conditions, then you get these. So yes, there are changes that can be observed. Those two spectra are not identical, and then these changes reflect changes in the reagent with respect to the protein in the first state with respect to the protein of the second state. And that can be followed. It gives you information about where the reaction has taken place and the conditions that has happened, and the new structure that you might have produced after reaction has taken place. So an example of reaction-induced infrared difference spectroscopy is, for example, photolysis reactions. Here you've got an example at the top of the photolysis of cage ATP. In its cage state, it is attached to this nitro compound. The ATP does not react with a protein. When you photolyze your cage ATP and ATP is released, then it is free to react with your protein of interest. And in this example, this spectrum here is the behavior of calcium to ATPase sample. So the thick line in the spectrum is the cage ATP with no protein. So that's how the spectrum looks like if you just take a picture of, infrared spectrum of the cage ATP with a nitro compound attached. And the thin line is once you have photolysed the cage ATP and how it behaves when there is ATPase in the sample. So again, as you can see, the two spectra are very different, and you are able to identify in different areas of the spectrum where that ATP might have attached to the protein and in what quantities. And so it is able to follow the reaction with protein and without protein, and what happens when the ATP is released, and where in the molecule, where in the protein that ATP attaches. So again, we are looking at the relationship between the protein structure and its function. So this is all for the video. I hope it was useful, and I hope it has given you an overview of what you can do with infrared spectroscopy to study proteins. Please do contact me if you have any questions, and thank you for watching.